platform for scientists to share their research and innovations with the global LRIG community. All our events are free and open to all. If you would like to organize or to present at a future scientific meeting, please email your proposal to talks.phl@lrig.org. I'd like to put out a special thank you to Lorena, who's our host today, to Sharon and Ron. Ron will now introduce our speaker, our first speaker for today. On to you, Ron.
Thank you. Today, we'd like to welcome Rwik Sen. Dr. Rwik Sen is a field application scientist at Active Motif, Inc., and with his doctoral and postdoctoral training in epigenetics, transcription, DNA repair, and developmental biology, which aligns with Active Motif's portfolio of high-throughput epigenetics assays like ChIP-Seq, Hi-C, ATAC-Seq, RNA-Seq, protein interactome profiling, etc., with the PIXUL Sonicator. Today, his presentation is entitled Advances in Sonication for MultiOmics Applications with PIXUL. To you, Rick.
Thank you, everyone. Thank you, LRIG, for the opportunity to present today, and to all the attendees for their time. So my name is Rwik Sen, and I'm the field application scientist at Active Motif, and today I'll be talking to you about our high-throughput multi-sonicator called the PIXUL. So PIXUL stands for pixelated ultrasound, and as I show you some of the results that we've obtained with the PIXUL, I hope I'll be able to convince you that PIXUL can fit a lot of research needs across a wide variety of fields. So a brief history about myself is I did my undergraduate in biotechnology from India, and then I came to the US for my PhD in SIU Carbondale, where I studied molecular biology. And then I did my postdoctoral training at the University of Colorado, where I also had a fellowship from the American Heart Association. And apart from research, I am also interested in, and I practice these very actively, mentoring, teaching, outreach, and I'm a big advocate for inclusion in STEM, and I also do science writing. So currently I'm a field application scientist at Active Motif, and this is how Active Motif looks like. It's in a very scenic location in Carlsbad, California, and you are very welcome to connect with me on LinkedIn here. And if you want to know more about my publications, you can find them here in PubMed. So a brief history of Active Motif before I go on to PIXUL. Active Motif was founded in 1999 by Joe Fernandez, who was the co-founder of InVitroGen. And this is how we progressed through the years until in 2020, we launched the PIXUL, along with very interesting COVID-19 portfolio. Now, Active Motif is situated in Carlsbad, California, where the headquarters are, but we also have offices in Europe, China, and Japan. And we cover almost all of the people from the entire world. So you can read more about our story in this podcast link here. Now, we have a lot of products that's directed towards epigenetics, but we also have products that cover a lot of other research areas in the biomedical field. And we offer end-to-end epigenetic services like them listed here, and I can talk to you more about them offline. And all these customer publications, peer-reviewed publications, have listed our products and services. So now coming to PIXUL. Why PIXUL? So PIXUL was developed to address some of the issues that people usually face with sonication. So for those of you who are not familiar with sonication, it is a process which you need to break open biological material, cells, tissue, or anything that you need to extract content from in order to do your experiments. And one of the principal purposes of sonication is to cutting the DNA or chromatin into small workable pieces, because as you know, DNA length is very large, which is not very easy to handle if you are trying to look at specific regions of DNA for high resolution, a very small base pair ranges. So just to give you a summary, this is how the cell looks like schematically. The blue is the cell membrane, then the pink here is the nuclear membrane. Inside the nucleus, you have your chromosomes. If you look at the finer structure of the chromosomes, then you'll see that there are fundamental units of the chromatin, which is called a nucleosome. So the nucleosome is actually an octamer or a group of 8 proteins. Those proteins are called histones, and around them the DNA wraps around, and it exists like that in that condensed structure. Because DNA is so long, it has to be condensedly packaged compactly inside the nucleus to exist, save space. Now, if you have all of them very close to each other, that's closed chromatin. If you have them far apart, they're open chromatin. So open chromatin is more accessible to transcription factors. You have active transcription, all the genes that are highly transcribed. You get the output as RNA, and those are translated into proteins. If the chromatin is closed, then you don't have accessibility to transcription factors. Your genes are silenced. So all of these events are happening inside the nucleus. People have different needs to study them. Some people are studying DNA, some people are studying chromatin, some people need to study RNA, and some people need to study proteins. Then there are different modifications happening on the DNA, like this ME here, that's methylation. A lot of different modifications happen on the histone proteins. They have tails, so on the tails of the histone proteins, and people need to study that. So for all these different reasons, you need sonication. And to help with your sonication, we have brought you PIXUL. So for those of you who have been doing sonication, you may be very well aware of all the problems and the bottlenecks in the sonication workflow. I was doing a lot of sonications during my PhD because I did a lot of ChIP experiments using yeast cells, Saccharomyces cerevisiae. So the first bottleneck was time. In order to do a sonication, to set up the sonication and to get everything ready and done and go to the next step, it would take almost the entire day. Then the other one is the sample transfer. So every time you do every time you have to set up the sonication system, you have to grow the cells and then manually transfer them, the cells, you harvest them, and then you transfer from tube to tube, you do different assays. And in the end, by the time you end up going to the sonication system, you have done few batches of transfers, and that has led to loss of your protein. Now, it may not be a concern if you are growing yeast cells, which you can grow lots, but if you are working with samples which are limited, like patient samples, then you can't afford to lose them. And then you also need a lot of labor, and which is not always easy to get. Then setting up of the sonication system, that itself is an elaborate process. You need to degas the water, and then you have to worry about consistency, and then finally the downstream analysis, again, you do transfers. So PIXUL helps you address all of these bottlenecks. First, you don't have to do manual transfer anymore because you have these 96 well round bottom plates, which are tissue culture treated, TC treated, and you can grow your cells in here. When you're ready, you just change the growth media from here. You add sonication buffer. If you need to do fixation, you just add formaldehyde, do your fixation, add glycine, and then switch that out, add sonication buffer, put that plate in PIXUL. So it's the same plate that you have your cells growing in. You put it in PIXUL without having to change anything. PIXUL setup is around 5 to 7 minutes. By that time, the machine is ready to run. Then the shearing process itself is very quick. The entire 96 well plate can run in 30 to 36 minutes if you are sonicating for gDNA or chromatin. If you are sonicating for RNA or protein, that's much less. It's like 5 minutes. Yes, it is 5 minutes. And then downstream transfer, depending on what kind of assay you are doing, it's quite easier compared to doing from tubes to plates and things like that. So you're saving time, you're saving labor, and you're saving your samples by not having to transfer from plate to plate. One question we often get is this plate proprietary? No, it's not. You can get this directly from a vendor. And so that means they are very inexpensive. A very cool thing about PIXUL is that it allows you to program 12 different columns on this 96 well plate. So this 96 well plate is a standard 96 well plate, round bottom plate, which has 12 columns and 8 rows. Each of the 12 columns can be programmed differently in PIXUL. That means you do not have to have the same sample across all the wells. You can do different kinds of samples all in the same batch and just program it differently, and you get everything done in one run. Also, you do not have to fill the entire plate with samples. If you have few samples, you can just fill those few columns and program that. So PIXUL was first developed and reported in this Nucleic Acids Research 2019 publication as a collaboration between Active Motif and the University of Washington, Seattle, where the manufacturers are. So from from the University of Washington, Seattle, there's a company called Matchstick Technologies by Dr. Carol Baumstick and Dr. Tom Matula. And then we also have Greg Darlington from the engineers, shippers and crew, who all helped to assemble the PIXUL. And PIXUL looks like this. As you can see, this is very sophisticated and portable, as you can determine from the dimensions and the weight. This is all that is there to the equipment. There is no additional water bath, no computer, nothing. So this really has a very convenient footprint. And since it's portable, you can just move it around anywhere in the lab. You do not need to place it in a cold room because it has inbuilt chilling capacities. And you do not need any additional sound protection equipment because it is very quiet. You won't even know it's running. PIXUL operates on the principle of sonication that is based on the principle of cavitation. So cavitation is when you have a series of compression and expansion going on in bubbles in a fluid that ultimately leads to implosion of the bubbles, releasing energy. The energy that is released in the form of heat, light, that is converted by transducers into ultrasound. That ultrasound is focused on the samples using lenses. So that ultrasound that's focused on the sample that causes the sonication of the sample. Now, a very important feature of PIXUL is that these bubbles, ultimately the bubbles that are imploding, they are generated to be very uniform in size and very small in size compared to other technology that operate on similar principles. So because the bubbles are generated at such uniformity, homogeneity, and at such small perfect sizes, that PIXUL scores over a lot of other technology in terms of consistency. We have heard this across the field from different kinds of users, be it DNA, RNA, chromatin, protein. They have all been very happy with the consistency of PIXUL. So that's where this consistency comes from. If you can see this video, then it looks like this, the bubbles expanding and compressing, ultimately giving you the energy. So I'll show you some results, which hopefully will convince you that PIXUL is consistent. And if you are to have PIXUL for an evaluation in your labs, then you will be convinced of these other features, that it's simple, fast, and flexible. And also it's affordable in the long run because the consumables are really quite inexpensive compared to others. So as I mentioned, PIXUL is very sophisticated. It just has these two switches and this touchscreen. That's the main user interface. Everything's done through here. And your plate goes in here. It sits right there. And this is a chamber for coupling fluid. So the bubbles that I was talking about, those bubbles are caused in the coupling fluid. So the coupling fluid is a viscous fluid. It is antimicrobial. So you do not have to worry about biological contamination growing inside the machine. Once you pour coupling fluid in, you never need to flush it out. You just keep topping off as and when it runs a little low after some number of runs. So there is no need to worry about contamination or anything else. This touchscreen helps you to program PIXUL, and as you can see, you can select columns at your discretion, and you can assign the parameters. So these are some frequency parameters: pulse, PRF, burst rate, and the process time is here. We have optimized all of the frequency parameters to be exactly these. Sometimes we can change them, and that's fine, but mostly we have simplified the process by only having you change the process time as and when you need it. So you don't have to worry about like changing all 4 parameters, just keep changing the time. And I have full Excel files of what kind of samples work with what kind of parameters. So if you are interested, I can provide them to you. Temperature is maintained here. And so once you put your samples in, you circulate for the temperature to come down, and that was the time I was showing you earlier, that's 5 to 7 minutes. So this is your room temperature. When you press circulate, your temperature comes down to colder temperature, and then you start circulating, sonicating. So that time, that is the only setup time, and that is like just 5 to 7 minutes that your machine is ready to run. Then you enter your parameters. When you run, whatever highest time you have entered, that will be your total runtime. So if you were running this screen right here, your run would be done in 20 minutes, and all your samples would be processed by then. So PIXUL has your plate sitting in this area right here, and this area, it has the lens array, the lenses that I was talking about that focus the sonication energy to your samples. And this region has 24 lenses. So one lens is powering 4 wells in each column. So the work is highly distributed across 24 transducers, and the 24 transducers are sharing it, so each of their lifespan is much more increased. They are not stressed. It's not one transducer powering your entire plate. These are some volume requisitions for PIXUL to run, and this is how your energy is processed, focused. Now this video, if you can see the springs coming out of here, this is the internal anatomy where you see columns are getting sonicated in pairs. This is done to minimize the heating. So the coupling fluid not only gives you energy bubbles for sonication, but it also keeps your temperature down to the proper level. Now I'll quickly go over some results, and I can talk to you in more details about these. I won't take too much time going into experimental details, but just to show you what PIXUL can do. So if the paper that I showed you, there we have done these experiments, rather the authors have done these experiments, where they've grown cells, fixed, quenched, and added shearing buffer all in the same plate, then sonicated. Tissue is interesting because homogenization had to be done outside, but now we have good news that tissue can also be sonicated directly in the plates with the buffer. So you don't need to do anything outside. These are some quality control data. You see all 96 genomic DNA samples, they have sonicated with extremely high consistency here. The starting material does not affect the sonication efficiency. Only the time does if you keep everything else constant. So you can see 20 microgram of DNA all the way down to 0.5 microgram. They are all giving you the same sizes after sonication. A good question we always get is well-to-well cross-contamination. No, we don't have that. We have ruled that out by PCR. So PCR is very sensitive. If there was contamination, you would detect that. Size ranges, we have got 200 base pair to 13, 14, 15 kb, and we are still expanding on both ends. We have high consistency. You see all 96 samples sonicated very nicely, overlapping. The parameters, irrespective of whatever DNA you start with, you see all of them are very highly consistent after sonication. Same consistency for if you grow cells in the wells instead of adding genomic DNA. So here I'm showing you 100,000 cells to 1 million cells per well, but now we have stretched both the ends of this range. We have done 10,000 cells and 5 million cells, and all of them show us this consistency here. Compared to other technology, you'll see in the paper, it's shown that PIXUL variation is much, much less. So here are some more experiments where we have shown that using PIXUL, you can generate results which are true. So in a usual traditional experiment, if whatever you will see, you'll see the same thing using data generated by PIXUL, because you may have doubts whether a new technology will give you the true results or not. So those things have been shown in the paper. Basically, we have shown that different regions of the same gene can be studied perfectly. Different genes can be studied, and different time points of serum induction or any induction or anything can be studied here. ChIP-Seq is another high throughput assay for people who are looking into those kinds of chromatin readouts. So in ChIP-Seq, we are looking at the genome-wide occupancy of specific transcription factors or histone marks of our interest. So it has to be very high throughput and very consistent, and it's very expensive experiment, so you cannot afford to make mistakes. And For that, you need very high consistency sonication, because sonication is one of the most important parts of a chip assay. So using PIXUL, we have been able to get very high consistent ChIP-Seq data. You see the profiles are very nicely shown here. They're aligning. So for people who are not familiar with these, so these are all histone marks that we are looking at, histone modifications, and we are seeing where those marks are enriched versus where they are not present. So you see certain marks are present in certain regions versus they are not present in other regions, which is as expected. Then tissue processing, you see we have done tissues ranging from 1 milligram to 30 milligram, and all with very unique profiles and very nice no backgrounds. ChIP-Seq data generated by PIXUL correspond with ENCODE samples. So ENCODE is the consortium which globally people follow, and ENCODE, whatever ENCODE gives, that is true, and then ENCODE and PIXUL generated chips match. So that is really, really exciting. If you want to look at RNA, we can do so, and RNA is also very nicely consistently sonicated. We have compared ChIP with RNA. So two different readouts, but they ultimately tell you similar things. So if you have occupancy for RNA polymerase 2 in ChIP, then you should see RNA expression. And exactly that is what you are seeing. You're looking at different tissue-specific genes, and the results are very nicely matching. You can also put the same sample in the wells and add different kinds of buffers to look at different readouts from the same samples. And that is exactly what we have done here. We have looked at ChIP results for RNA polymerase 2 occupancy. We have looked at RNA results from the same cells, and then we have looked at protein, and all of them match nicely with each other. You see a 30-minute signal, 30-minute signal, 30-minute protein. There is a difference between 15 and 30 versus from here to here, and we know that's because of the RNA polymerase 2 pausing before starting the transcription. So that's why there is a lag. People in proteomics field have used PIXUL in workflows of MultiOmics, LCMS, and other assays that has led to increased scope and sensitivity. So they have also been using this in different assays, and because of confidentiality, I cannot mention their names or anything, but I'm happy to talk more about the proteomics groups offline. Some kits and consumables, which people ask us about PIXUL, you really don't need our kits and consumables to run things on PIXUL, but we do have them if you are new to Sonication and if you want to try. But our customers have used both their kits, our kits in PIXUL for comparison, and have not seen a difference with the PIXUL generated results. So you can use your own kits, and that should be fine. Evaluation program. We are having PIXUL evaluation at your labs for free. We ship you a PIXUL. Everything's done remotely because it's so simple. I help you to set up the PIXUL, run protocols and everything, and then we just encourage you to share your results. And if you like the PIXUL, then we are happy to help you with the purchasing process. And we offer you free consumables for the first several months, or however we can help you. So I hope I have convinced you with some of these aspects of PIXUL, and I'll be happy to talk to you more about this and connect with you. And with that, I would like to thank everybody at Active Motif and Matchstick and our customers who are collaborating with us and providing us a lot of data. So we have some t-shirts and stickers that Active Motif is famous for, and please follow us on social media. And with that, I'd like to thank everyone for your attention and take questions. Thank you so much.
Thank you, Dr. Sen. There are a few questions. First of all, does PIXUL have temperature control?
Yes, PIXUL has temperature control. So we have inbuilt chillers, and when I was showing you the slide where we do circulation before the start of the sonication, then it brings PIXUL down to a certain temperature that we need, and then you start the sonication, and it maintains and even cools down further.
There's a two-part question. One is, in the video you show the mist coming out of the wells during sonication. Yes. Right. Why doesn't this cause cross-contamination, and how is that prevented?
That's a great question. So what I showed you here is what you will not be able to see in the actual machine because it has the lens array on top. So you will. So this is just to show you how the functioning is happening, and your samples are in the microplate. So the bubbles are hitting the bottom of your microplate on the outside exterior surface. They are not coming in contact with your samples in any way. So your samples are not getting cross-contaminated, and the plate is being sealed using a plastic seal that you would use to seal a RT-PCR plate. So it's just a regular seal from any vendor that you can get. If you are working with proteomic samples, for sensitive samples, we have the option of non-adhesive films where your samples, they may get in contact with the film, but it won't have adhesive, so it won't be contaminated. We have those options for you as well.
Excellent. I'll just a couple more questions. What power input are you using, like the frequency?
The frequency, so we have three frequency parameters here. And the first one is called the pulse, that is 50, and then the PRF is 1 kilohertz, and the burst rate is 20 hertz. So these are some of our frequency parameters.
And I guess it's a question that I had that is related. So you have 24 transducers, and it looked like you did not have variability when you ran the standards across the entire 96-well plate. That's good. Does the volume of each well, if there's a difference in the volume, does that create a well-to-well difference?
No, it does not create a well-to-well difference because we have, so these are the volume ranges that's recommended. Now, it may create a difference if your concentration is different in different wells. So for that, you may need to optimize the time for the sonication. But unless there is anything different in your like sample to sample, well to well, PIXUL itself doesn't give you any heterogeneity in the output.
Okay, excellent. And the last question is, are there bubbles formed from the gases dissolved in the shearing buffer?
The bubbles are, so your bubbles are not coming in contact with the shearing buffer because your plate is completely sealed. Bubbles are just hitting the plate from outside.
Excellent. Well, we thank you, Dr. Sen, and we can pass the ball now to Gary.
Thank you very much.